Thank you. Thank you, Stuart, and the organizers. It's been a really wonderful meeting so far. Enjoyed all the talks. But I'm going to talk about something different. Plants. We had a discussion, do we need brains for consciousness? Well, let's explore something differently. So I'm a photobiologist, and I work at Carnegie Institution, which is located at Stanford. Let's see. Trying to see which one is the forward?
This? Yeah. Sure, it's on. Oh, it's the other way.
Oh, that's
You're backwards.
perfect. Thank you.
It's just the arrows backwards.
Okay.
Hold it towards the back.
Okay, so let's start by asking a question. We talk about consciousness, but the word consciousness, many people have different types of meanings for it. So we'll explore a little bit there. But let's ask the first question. What is the difference between a non-living form and a living form? I would say a non-living form is subjected to its environment. Whatever is happening to the rocks, to the ball, to the sand, there's going to be a reactivity, either at the physical or the chemical level. A living form that is alive is responsive to its environment. The dog is going to play with the ball. The child is enjoying the ocean. So if that's true, and we look at a picture like this from Yosemite, and I ask several people, what do you see here? Mostly I get answers. I see a waterfall, I see clouds, I see mountains. On the right-hand side, I see deer, I see snow. Nobody says that I see plants. This is known as a phenomena, plant blindness. Actually, humans have it. We never see plants. But plants are very much living. They are alive. So if plants are alive, let's take a step back to how we are related to them. Okay, they are all around us. We depend on plants. Plants don't depend on us. So if you look at the evolutionary process, the Earth is, let's say, 4.5 billion years old, right? And about 3.8 billion years ago is the first evidence we have of life. That's not a very long period. It's less than 1 billion years from Earth to having arrival of life. So what we don't understand, I would say, are two big questions. How did life arrive or appear on Earth? And what is the experience of life? Those are the two big questions that we're trying to answer. So if we start to follow along this tree, the first fossil trace of eukaryotes is about 2.7 billion years. And then we get to this branch. You can see this branch, one going left, one going right, the green and the red. This is the point where plants and animals we know had a common ancestor. Why we say that? Mainly because of both plants and animal cells have mitochondria. And mitochondria, as we know, have prokaryotic origin. So we look at a protobacterium that was assimilated into an initial cell that was ancestor to both plants and animals. So there is that connection. Now what happened after this? Plants also acquired cyanobacterium, which has the ability to do photosynthesis. So plants started to do photosynthesis, and we took a slightly different path, the animals. Now, there is evidence of multicellular eukaryotic fossils about 0.6 billion years ago. This is where we are today. But just to show you the scale, that's when the dinosaurs disappeared, 65 million years ago. So life goes back quite a ways back. So why am I showing this? Well, the logic of developmental process is similar in life. It is survival. Any organism, they're using different molecules and arrangements of different molecules, but there are protein domains. There are regions that we can trace back all the way to the old prokaryotes. So protein domains and many things are very highly conserved. We do believe that plants do represent a proper comparison to animals and can be used to study pathways and mechanisms. So this is an anesthetic being applied to a human. We can breathe, talk, walk. Plants don't breathe, talk, or walk. But what do? They respond to the environment. And they grow differently when they're subjected to complete darkness or when they're introduced to light. So in my lab, we apply anesthetics to plants. And it's not the first time. The very first anesthetic experiment that was published was in 1878, I believe. So people have been trying anesthetics on plants for a long time. But most of those studies have always been about looking at growth or response of mimosa pudina, which is also known as touch me not. So the plant that closes when you touch it, and those plants stop growing or stop responding. But it's not very well known. We don't know how anesthetics work in the first place very well. So can we use plants to understand how anesthetics work? This is not a talk about whether plants are conscious or not. This is about exploring pathways for anesthetics and psychedelics, but using plants. So let's look at that. So I mentioned that plants grow differently in dark and light. So just to answer this question, we had to pick some mechanism, some function. Being a photobiologist, I like light, so we start with light. If you look at light, you can see the visible spectrum is a very narrow band. If you look at all of the radiation that we can measure, the visible spectrum is in the middle and it's very narrow. It goes from 400 nanometers to 700 nanometers. We're going to focus on these particular monochromatic wavelengths, blue, red, and far red. Plants respond to, they have developed specialized photoreceptors to absorb these particular wavelengths. One thing that is very well known is phototropism. Now, phototropism is bending of seedlings or plants towards light. And I don't know if you knew this before, but it is very well established that plants only bend towards blue light, not red, not any other. White light has blue light, so they will bend towards white light. So if we put an LED, blue light LED, to the right, just looking at the left panel, and we apply different amounts of isoflurane, anesthetic, we can see the control, which is no isoflurane, is very nicely bending towards where the blue light was. As we start to increase isoflurane, we see that that response is lost. Now, if we wait one day, and these seedlings were left underneath a white light source, which was on the top, you can see that, again, the controls are, next day, they're already bent from the previous blue light, but now they're going up towards the white light. And you can see that even the ones that did not respond to blue light are now recovered. So with anesthetics, we were able to show that the seedlings lost the ability to phototropism, but it was transient. Following day, there was recovery, and it was dependent on the concentration of isoflurane. Now we can measure this. So if 180 degrees is straight and bending is 90 degrees, you can see that in the graph there, 90 degrees is the bending, and then as we increase the amount of isoflurane, there's less and less bending. So it's dose dependent. So this is something that has never been done. Many times plants have been subjected to anesthetics, but this kind of dose dependency has not been performed. What's interesting about this is that it's very similar to human and animal doses for loss of consciousness and going towards surgical anesthesia. So you can see that we are right about between 0.5 and 0.8, where transition from loss of consciousness to surgical anesthesia. And so the volumes of anesthetic are very similar that are being used in other organisms. Okay, so this is good, this was exciting, but there was one problem. Giving an anesthetic could have many effects, could damage the membrane, could do so many things. One question was, phototropism obviously is monitored or controlled by phototropin, which is a photoreceptor. So one question was, did we lose the function of the photoreceptor? Perhaps that's why there was no phototropism. Well, if we use a mutant where there is no photoreceptor, then we won't get phototropism anyway. So I had to go to a slightly different system. And I'm going to explain this because this is going to lead to some very interesting discovery. So plants grow very differently in darkness, as I mentioned, and also differently in continuous red or continuous far red. You can see that in darkness, we call that process etiolation or scotomorphogenesis, whereas growth in light is photomorphogenesis, both red and far red. One thing you can notice is not only the hypocotyl is shorter in light, but cotyledons, which are the first leaves, they are open. But in far red light, there is no greening. So you need either red or blue light for chlorophyll synthesis. Okay? So if far red light, plants will not green, they will stay white, and they will not be able to photosynthesize because you need red or blue light for photosynthesis. So what this is telling us is that light carries information. Right? So different colors of light, plants have completely changed their developmental program based on what type of light it is. So this information can be color, direction, duration. All of these things are sensed by the photoreceptors, and plants behave differently. They, in fact, absolutely completely change their developmental program, as we can see here. And this is so precise that we did a microarray a long time ago, and from the microarray we could see repressed genes, or genes that were more activated. We could order mutants. This is the best thing that we can do with plants, harder to do with humans. We can order plant mutants, and single genes missing. And you can see those differences. They look small, but they are always repeated the same way. So we can study gene function very easily using seedlings. So this process starts with photoperception. So this on the left side are the controls. You can see the wild type grown in red and far red, and then you can see again dark. And all the genes, single mutants, you can see how differently they are. The important thing is on the right-hand side where you can see phyA, phyB. In red light, phyB, phy stands for phytochrome, which is a photoreceptor. When phytochrome is missing, the seedling is unable to see red light. See, it's tall like it is in the dark. When phyA, phytochrome A, is missing, the seedling is unable to see far red light. So these photoreceptors are very important. So light is perceived by a photoreceptor. The process starts with signal transduction. There is gene expression, protein modification. The whole process regulates growth and development under specific conditions. So we applied this mechanism to study isoflurane. Now in this case, we included phyB, which is on the right-hand side. What you can see in the photograph is that when we increase the concentration of isoflurane with the wild type, you can see a greater effect. But isoflurane impacted the wild type as well as phytochrome mutant similarly, which means that missing the photoreceptor did not have any effect on the impact of isoflurane. So the question was isoflurane directly affecting the photoreceptor? And it doesn't seem to be. So we can see again quantification of hypocortical length. You can see red light, Columbia is the wild type, and phyB is the mutant. So under red light, you can see isoflurane having similar effect. And in dark, you can also see that in both cases, isoflurane is having an effect. But when we quantified this, something interesting happened. If you look at the percentage of change, we can see that the same amount of isoflurane, 0.5, as we were looking at before, has a greater effect in red light. In the dark, it had lesser effect in hypocortical growth. So now there is a differential of anesthetic acting upon a seedling, acting more strongly in presence of light and less effective in darkness. And this is very interesting because in the dark, as the seedlings grow, the majority of the differentiation, so there is no differentiation, actually development is cell elongation. So the development in the dark is only cell elongation, whereas in light, there is cell division, there is lots of gene expression, and we're going to look at that a little bit. So it's interesting that the basic functions of cell elongation are more intact. Now, if you imagine a patient laying in the hospital, they're still breathing. There are some basic housekeeping functions that are maintained. The ability to respond to the environment is affected. That's what we are seeing here as well. So the ability to respond to red light is more affected, whereas cell elongation, some of the basic functions are less affected. So what's going on here then? If you look at, again, wild type and phytochrome A and B mutants, we can also see that there is a difference in cotyledon development, and basically, this whole system is so well studied. I've been studying this whole system for close to 25 years, and all the genes are known, everything is known about it. So how does it work? And this is where the discovery was made. So let's follow along with something that's very well known. So I'm just going to go over a couple of things. We'll start with this protein complex known as COP1. COP1 stands for constitutively photomorphogenic. It's interesting because constitutively photomorphogenic means that even when it's in the dark, it thinks there is light. How does that happen? If we take all the light, we have the photoreceptors in plants for far red, blue, and UV. I mentioned phyA, phyB, and there is also phototropin. So when light comes on, light suppresses COP1 complex. When this COP1 complex is suppressed, it releases its effect on downstream proteins and genes. Now, COP1 is an E3 ligase. So what it does is that it sends all these proteins for degradation. That's all it does. It will take all those proteins that are downstream and target them for protein degradation. Now, when COP1 is missing, those proteins are not degraded. You can see on the left-hand side, when COP1 is present, it activates scotomorphogenesis or cell elongation. When light suppresses COP1 complex, all the lower proteins are activated, and you get that completely different developmental program. So, what if we remove COP1? We mutate COP1, this suppression is gone, let's remove the light as well, and all of this is gone. Now that pathway is active, and it is in dark. We should get this, right? And that is what happens. So if you look at the wild type, there is etiolated plant grown in light. You can see COP1. In dark, it grows as if there is light. So you don't even need light. So deetulation, which is removal of dark growth, can proceed when both light signal and the off switch are not there. Now, survival depends on light. You can see that the COP1 seedling in dark is not green because chlorophyll biosynthesis requires light. But this tells us something, that the signal, extrinsic signal, is regulating development and is directly linked to gene expression, protein modification, all these processes that are happening. So let's look at another program, and this time we'll talk about microtubules. So one of the things that also plants do is that they open and close their stomata, which are present on leaves. You can see guard cells. There are two guard cells. They're around a pore. The pore is the stomata. And I created these lines in 2014. These are GFP-TUBULIN expressing in wild type as well as COP1 mutant. Now, COP1, as you know, does not care whether there is light or not. So if you look at the wild type, you can see microtubules forming these very beautiful radial structures within guard cells. In plants, microtubules are very well studied. As we just heard before, microtubules have also been directly linked to many functions. Opening and closing of stomata, they provide the cytoskeletal structure to guard cells. And they reorient, they assemble, they disassemble. Both Rick and Cassandra showed beautiful pictures of how microtubules can grow and retract. And so these are directly related to functions that are important for plants. So if you look at on the right-hand side, you can see that the stomata looks slightly different. That's because in the wild type, stomata will open or close depending on whether it's dark. They close in the dark, they open in light. Whereas in COP1, they never close. They're always open. And so they're stuck in this radial structure. And so I thought that would be a good mutant to study for further studies with isoflurane. So here's a stereoscope image of the wild type and COP1 mutant. You can see that we can see beautiful GFP-TUBULIN. Microtubules are everywhere in the plant. But now we start to look at these two with isoflurane added. For 1 hour, you can see 0.25% and 0.5%. The top one is wild type, and the lower one is COP1 mutant. If you look at the top one, there are two plants, plant 1, plant 2, and at 0.25%, we see beautiful arrangement of microtubules in the leaves. But now when we go to 0.5%, with the confocal microscope, we cannot see microtubules anymore in the wild type. But they are present in COP1. So COP1 is able to withstand 0.5% of isoflurane. So we found a mutant that no longer responds to anesthetic, like the wild type. This suggests that isoflurane is not simply affecting the membrane, it's not a passive effect, it is actually involving genes, and there's a pathway. So this is exciting. Now, Travis also showed this yesterday, and when we saw these blobs, we were trying to understand why these are blobs in the wild type, and yesterday we saw these blobs as well with isoflurane. I think it was with psychedelic, one of the psychedelics. And so these blobs appear in the wild type. And we are trying to see whether they are just GFP or most likely GFP-TUBULIN, because GFP-TUBULIN is constantly synthesized. So we are also looking at recovery process to see if the microtubules come back after treatment with isoflurane. But now if we look at the COP1 mutant, none of that is happening. So COP1 mutant is resistant to the effect of isoflurane. Now we're going back to MAPs. Plants also have MAPs. And some of them also have similarity to tau. This is a particular map, WDL3, and it's been known for a long time, since 2013, that WDL3 binds to microtubules and it does stabilize, like map 6, 7, 9 you said. So this does stabilize microtubules, and it beautifully highlights microtubules in plants. You can see that it's present in light, and it's degraded in the dark. Guess who degrades it? COP1. So it is also shown that COP1 degrades WDL3. So our working hypothesis is that COP1 degrades WDL3 MAP in the dark. WDL3 stabilizes microtubules, and in absence of COP1, WDL3 accumulates, and so COP1 mutant shows a reduced isoflurane response. So access to microtubules is blocked, which suggests that isoflurane may bind microtubules directly. So we are testing this, and one thing is all of this study is in vivo, and these plants are very much alive and living and doing their normal things. So we are able to study, because we can put GFP-TUBULIN into plants. It's very hard to do that in human brain. So it makes it easier to study functions in vivo, or pathways in vivo. So isoflurane anesthetic interacts with microtubules to suppress environmental signaling response. Isoflurane loss of consciousness, is that a result of inhibited signal transduction? Do plants and animals share similar mechanisms? Then what is consciousness? So I'm going to just propose something. In my mind, consciousness is a classical signal transduction mechanism that drives individual potential. And it has to do with interrelatedness with spatial information. So there is information in space-time, and we are just like light. Light is a quantum photon, and so plants are able to take that information and change their developmental program based on the information that's present. And it shapes the degree of fitness and evolutionary succession at microscale. So I thought about this a lot, and I have a theory. I call it theory of spatial relativity, and this theory aligns with OR, mainly because there aren't that many other theories of consciousness that explain quantum level interaction, and basically going from physics to biology. So what we need to do is go from the understanding of the physical universe to the understanding of biological responses. I have a showcase talk. You're welcome to watch it on theory of spatial relativity. So just to finish, consciousness then is a process, in my mind, similar to photosynthesis. Plants do photosynthesis. They utilize information, light energy, water, air, in that case carbon dioxide, and they produce stored food, store their food with a degree of efficiency. So different plants will do photosynthesis in different ways. Whereas humans, they develop consciousness. Maybe plants are conscious, I don't know. But the consciousness in that case is a process which utilizes hidden information, something we cannot measure. We can measure light. But there is, at quantum level, there is information, then also utilizing water, air, oxygen, let's say, and components of other components, and then respond to the environment in varying degree of self-awareness, which is related to efficiency. So in that sense, if you think about the evolutionary part again, where the plants and animals split, plants excelled in developing photosynthesis, and we excelled in developing tools for consciousness. So if you buy this, at least I can say that plants develop processes to produce food for all life on the planet. Animals develop processes to think and the ability to rationalize. Well, plants are doing a very good job producing food for us. How are we doing? Right? So I have a podcast as well. We have wonderful guests. I'll just play this. We work on, if you can please start the video. I don't know if I can from here.
Two things that motivate me are the beauty of our environment, the uniqueness of our planet, of course, but also the people I've met, and the fact that people do want to play a stewardship role.
Welcome to Terra Science,
the podcast where reality matters.
I can actually say it. I'm going to be a scientist.
Excellent, you will be.
That's awesome.
Because I wrote it for people like you to get a sense of field biology, for women to appreciate the challenges.
If I try to do that, we're going to go down that rabbit hole.
I'm Rajnish, and I will tell you a little bit more about the food problem. It's a very complex and fragmented food system.
In that process of detection, that molecule momentarily is bound to us. It becomes part of us. So it's this incredibly intimate encounter that we have with the things of the world around us. And I simply hadn't appreciated that fact until I really began to look at it.
Maybe somebody will be inspired to also go into this field someday too. That would be amazing.
Well, I hope so.
I want to thank Bruce McIver and also Stuart because almost 2-3 years ago when I wanted to start working on this, they provided all the information we needed for the anesthetic treatments. And enjoy your rest of the meeting and stay safe during the storm.
Maybe one or two. Okay. Do anesthetics block photosynthesis?
That's a good question. So
Okay.
we haven't looked directly at photosynthesis, but my guess would be that it'll be transient again. And if we increase the anesthesia, it will definitely block it. But there might be a transient effect where it may slow down or stop, but then come back.
Okay. We better brief, please.
One is an enzyme, I think, and so do you imagine that the anesthesia is locking it to be active or inactive? Could you test the role of isoflurane on it in vitro and just understand how it's affecting to add to your model?
So the data that I showed was when COP1 was missing. So in the mutant, it wasn't there, and then isoflurane was not effective. So you're asking in the wild type, maybe it's acting through COP1. Which is also possible. We see COP1 controls many different functions. So when we look at isoflurane effects, they don't include all of the ones that COP1 regulates. So there does seem to be some specificity. So that's why we thought it might be downstream of COP1. But good question.
Let's thank Rajnish for a great talk and a great morning session on microtubules. Thank you.